Hello everyone. In this video I'm going to be talking about the liver and look more specifically at the functions of the liver. This is often taught at Key Stage 5. You tend not to go into the level of detail I'm going to go into in Key Stage 3 and 4. In 3 and 4 you mention the liver as being a store, if you like, of glycogen. So when you learn about blood sugar control and glycogen being stored as, or glucose being stored as glycogen in a way of controlling blood sugar, you learn really about the liver then, and you think about it in terms of digestion and the production of bile. But what we're going to do is talk about the liver and all its major functions. So let's draw what the liver kind of looks like. I mean, it's got this kind of characteristic sort of triangular shape almost. And in terms of coloration, well, it's almost, I'd say, slightly sort of brown-ish. So we're just going to shade this in here. A bit of a thicker. Just to give it a bit more of a visual. So we've got our liver here, it's got this sort of characteristic triangle kind of shape. So there's our liver, and we'll just go over the outline again, not the best shading. Now the liver is the largest gland in the body. So that's one thing I just want to make a note of here, a few little side bits here. It's actually the largest gland in the body, which not many people realise, and it uniquely has an input of blood from two separate sources. So you have, if we were to draw blood vessel here, now this is a descending aorta. So in another video I've referred to structure of the heart, and I've mentioned the aorta as being the largest artery in the body. Well, this is the descending branch of the aorta, and what you get is a supply to the liver from this descending branch here. So this is our descending aorta, and this branch that is going to supply the liver is known as the hepatic artery. Now it's important to mention this kind of like blood vessel structure supplying the liver before we talk about its function, because the fact that it has input from two blood sources is important. So blood enters via the hepatic artery, but there is also entry of blood from what's called the hepatic portal vein. So this blood vessel here is called the hepatic portal vein. Now, blood enters via the hepatic artery and the hepatic portal vein, and it travels what are called through sinusoids before leaving via the hepatic vein. So we're just going to put that in place there. So we have the blood leaving the liver through this vessel here, which is called the hepatic vein. So we had artery bringing the blood to this organ, and the hepatic vein carrying blood away from it back to the heart. Now the hepatic vein is continuous with the inferior vena cava, which will go and take blood back to the right side of the heart. So there's our basic kind of outline of the liver and its sources of blood. So we've got the hepatic artery and hepatic portal vein bringing blood to the liver, and hepatic vein carrying blood away. And I said that the blood travels through what are called sinusoids. Now, sinusoids are wide blood vessels, single-layered, porous, as in with holes in, and they don't actually have a basement membrane. So I'm just going to draw very quickly one out here. So let's imagine that we have from this side here, this is a hepatic portal vein. So we've got blood coming through this hepatic portal vein. What we find is it looks a bit like this. I'm just going to draw it very rough here. So you have these cells. We'll just draw a few in place. Here we have a source of blood, and this can be our hepatic artery. So you've got blood coming in from the hepatic artery and blood coming in also from the hepatic portal vein, and it's going to leave. So blood's going to leave through this hepatic vein. Now this whole structure here, all the way across down to here, this is what we call the sinusoid. It's a wide blood vessel, single-layered, porous with no basement membrane. Now these cells that I've been drawing, just put the nuclear into these cells. These cells that I've just been drawing here, they are what are called hepatocytes. That is the proper scientific name of the cells that make up the liver. So these cells here are called hepatocytes, and they will filter the blood. So these big cells are hepatocytes, and we have, just where I've drawn these in red, we have, I've not made it clear here, but there are pores or fenestrations, as they're sometimes called. So gaps within this sinusoid. In fact, we're going to reduce the screen size here, just so I can finish off essentially this little diagram of the sinusoid here. So we've got pores and or fenestrations, and what we have, and I'm going to draw these in small little purple dots located along those hepatocytes. You have this kind of structure. These little dots that I'm drawing are a very specialised type of cell. These purple dots are what are called Kupffer cells, and they're going to be used in the breakdown of erythrocytes or red blood cells. So we're going to have blood coming in, just for a visual, I'm going to put this in. So you've got blood entering from the hepatic artery, even though blood isn't blue again. For a visual, we've got blood coming into the sinusoid from the hepatic vein, and it's going to go down this sinusoid, this wide vessel, down here, and that's where the liver is going to perform its particular jobs. So you said this sinusoid is a wide blood vessel, single-layered, porous, with no basement membrane. Let's think about actually what the jobs of it are. Now, blood entering the liver has a variable nutrient content, and it may contain toxins that are harmful to other organs, like the brain. So clearly we need to filter and detoxify that blood. And that's what the hepatocytes do. So blood makes contact with the hepatocytes and it gets filtered and detoxified. So let's make a list on the side of the jobs or the functions of the liver. So, first of all, We need to regulate levels of particular nutrients in the blood because excess can be dangerous and can cause organ damage. That's critical. So that's the first job, if you like. That's what we need to say is an essential role of the liver, to regulate nutrient levels in the blood. And we're going to think of some examples of nutrient levels that we need to sort of regulate. So we're thinking here vitamins, we're thinking particular minerals, but also a key one is amino acids. So let's explain why I've written these three. Some vitamins or minerals are harmful in large doses and others may need to be stored in the liver for actual times of shortage. So that's why I've written vitamins and minerals. In fact, calciferol, or vitamin D, helps the uptake of calcium. So we're going to want to absorb and store that. So the hepatocytes will absorb and store calciferol, or vitamin D, to help calcium uptake. And also retinol, so vitamin A, that can be absorbed and stored and used to make other vitamins. So I said about amino acids. Proteins are made of amino acids. And when you have an excess of protein intake, and thus when they get broken down, an excess of amino acids, you can find that's actually harder to store. And excess amino acids can be toxic. So the liver, in a process known as deamination, will break down and destroy those excess amino acids. So regulate nutrient levels in the blood is a key job of the liver. So regulate vitamin level, mineral level, and also any excess amino acids you have there. One of the key nutrients we need to monitor is glucose. Now, glucose and salt can massively affect the osmotic concentration of blood. High sugar and salt can lead to increased water in the blood because it would lower the water potential inside the blood compared to surrounding tissues, so blood would move in, or water rather would move into the blood to compensate. So that's a bit of basic osmosis there. So that can be extremely damaging. High levels of glucose in the blood can change the sort of water concentration of it. So what the body does is produce, as you may know, it produces insulin when there's a high blood sugar. Insulin is released from cells of the pancreas and it stimulates those hepatocytes to absorb glucose and convert it to glycogen. So in another video when I've talked about blood sugar control, I said to lower blood glucose, we store glucose as glycogen in the liver. So that's what those hepatocytes can also do. So as part of regulating nutrients, I want to make an extra special point. In fact, and we'll just shrink down the screen again just so I can use more of the space. One of the key nutrients that is regulated is glucose. And glucose is stored in the liver as what's called glycogen. So we store glucose as glycogen in the liver. Let's think about some other jobs that the liver are involved in. So we've said about regulating nutrient levels of vitamin, mineral, amino acids, of glucose. Let's think about those hepatocytes at a more cellular level. Those hepatocytes, so let's put number 2 over here. Those hepatocyte, the liver cells, if we drew one bit larger here and got its nucleus here, there are structures inside that cell. I mean, I'm not drawing them overly well or detailed here, but there are structures that we call an organelle known as rough endoplasmic reticulum. Rough endoplasmic reticulum. It's a little structure found within animal cells. Now, these rough endoplasmic reticulum have what are called ribosomes on their surface. That's what all those little black dots are. And those hepatocyte rough endoplasmic reticulum, they produce plasma proteins. So this is an essential role of these rough endoplasmic reticulum. We get the production, so they produce what are called plasma proteins. Now these plasma proteins include albumin, globulin, fibrinogen used in clotting, so really essential. But those hepatocytes also produce cholesterol, which is important, and cholesterol is important in the production of bile. So here's our third key job of the liver. It's to produce what we know as bile. Now bile is produced by the hepatocytes, and it's actually secreted into narrow little vessels, what are called bile canaliculi. And then it gets carried into larger ducts to the gallbladder, where it's stored and where water is reabsorbed, and then bile is released into the duodenum, so the first part of the small intestine, via the bile duct when we're trying to digest food. So what bile does is emulsify food, particularly fats. It emulsifies fats to make them easier to break down. So the hepatocytes secrete this bile, it gets released or secreted into little vessels called canaliculi, and then passage into large ducts, where it then gets stored in the gallbladder. And then when it's in the gallbladder, it will then get released into the duodenum as and when required. So if I just go back to my original picture of the liver, and I drew the hepatic portal vein in, what I could have drawn in, actually just around here, and I'm adding it to the diagram at the very top of the screen, just located where I've drawn this sort of green dot. That is actually the gallbladder, and then you get coming from the gallbladder what's called the common bile duct, and that's this vessel I'm putting here, and the bile duct passes up into the liver. So what I've put in green on that initial diagram is actually the gallbladder and the common bile duct that goes into the liver. So the liver can produce bile. The liver is able to produce bile in the hepatocytes, and then it passes down that bile duct, the tube that I've just drawn, and it passes into the gallbladder. So this structure just here. So now we've put in green on that diagram at the top, the position of the gallbladder. And the final thing, where I'm going to end this video, is to do with the breakdown of the red blood cells, the Kupffer cells. So what you have, so we've said 1, 2 and 3. In fact, I'm going to finish by writing number 4 up here. So number 4, we said, is the breakdown of red blood cells, the fourth key role of the liver. So just to finish, I'm just going to explain a little bit about that. So red blood cells are destroyed after about 4 months or so by these Kupffer cells in the liver. So when a red blood cell ruptures, that hemoglobin, it sort of gets enveloped, if you like, by the Kupffer cell. It enters that Kupffer cell by a process called phagocytosis. So you get phagocytosis of hemoglobin by the Kupffer cell, and that hemoglobin then gets broken down. The globin, the protein part, gets broken down into amino acids that can be released into the blood. And the heme groups from the hemoglobin gets broken down into 2 separate bits. Iron, which can be used to make new blood cells in the bone marrow, and bile pigment. Now that bile pigment actually gets released or transferred to the bile, is released into the intestine and gets converted by bacteria to a yellow pigment known as bilirubin, which gives the characteristic colour to feces. So that's just, in a nutshell, the key functions of the liver. To regulate nutrient levels in the blood, to produce very important plasma proteins in the production of bile and the breakdown of red blood cells. Okay, hope all that helps.